I'm going to moderate this section with Dr. Janice Blair from
I don't know if it's
local. And if you are ready, please sit down, and we are going to start in right now. So in this section, it's focused on immunology, and we total have 6 speakers. And in this time, what we are going to do is when you are reached 12 minutes after your talk, we are going to raise our hand with some kind of papers, okay? And then when the speaker reach the last minute, the moderator will stand here. So you know it's time to wrap up. And then we will wrap up and have the questions, okay? And if we use all the 15 minutes, then we will not have enough time for any questions. So hopefully we can do it better this time. So the first talk is by Dr. Lisa Shibitz. Okay, she's going to talk about the B-cell depletion damage formation, granuloma formation in a mouse model of Coccidioidomycosis.
Okay. Thank you. Can people hear me through the microphone? Okay. Got it. Okay, so our background for this study is that, well, I'm wearing 2, so. Okay, is that better? Okay, so the background on the study is that, clinically speaking at least, B cells aren't considered particularly important for control of coccidioidomycosis. We use serum primarily to diagnose it, monitor regression, and B cells just haven't been considered that important. You can't transfer adaptive immunity with serum or B cell fractions given to mice. And humans with immunoglobulin defects do not have high rates of complications, including patients on rituximab, at least not according to what I was able to find in the literature. However, many years ago, McGee and Cox showed that B-cell deficient mice had reduced vaccine protection when given a recombinant vaccine and then challenged with virulent cocci. And then there's emerging data in tuberculosis in mouse and macaque models that there is a role for B cells in maintaining granulomas and preventing progression of disease. So in Mitch's recombinant vaccine challenge study in the MUMT mice, they showed reduced protection. And sorry, I can't even read the slides. There are some additional immune system abnormalities in the MUMT mice, but we're not going to talk about those. We also did a bulk RNA-seq analysis of chronic granulomas in B6D2F1 mice that were infected with 1038. And we found a great deal of message surrounding B cells and started to ask the question as to if they're not important, why are they there and what are they doing? So we designed this study to assess the role of B cells in primary granuloma formation in long stalk-dectin mice, which are black 6, but carry a longer stalk-dectin and have they're less susceptible, like a DBA mouse or a B6D2F1 mouse. And we looked at the role of the B cells in the chronic primary granulomas. So we gave 10 mice anti-CD20, and the other half of them got the isotype control. And we gave these mice the antibody every 2 weeks throughout the study. We randomized the animals before infection so there wouldn't be any selection bias when it came time to sacrifice them. And we started the antibody treatments 4 days before we infected the mice. We checked the B cell depletion 1 day before infection, and then we gave them 45 spores of the CP1038. We verified the ongoing B cell depletion before every antibody treatment and also terminally. We sacrificed the animals at day 54, which is essentially 8 weeks, and we tested them for cytokines and performed histopathology on them. For the cytokines, we closely dissected the lung granulomas and pooled and homogenized them. We also collected the spleens. And these were incubated without stimulation for 24 hours at 37 degrees with 5% CO2. And then collected them and tested cytokines using a 13-plex bead array for the mouse inflammatory panel. For the histopathology, we took the other half of the mice and fixed the lungs and the spleens in 4% formaldehyde, prepared histopathological slides. We stained 5 micrometer sections for routine H&E with PAS to bring out the spherules and make them easy to count. And then finally with an immunohistochemical stain anti-CD3 antibody to identify T cells. And we performed some quantitative microscopy using QPath. So none of the mice died in this study. And from an activity and general health level, their 2 treatment groups were indistinguishable. We did note a little bit of increased weight gain in the isotype mice compared to the anti-CD20 mice. So there may be a possible health effect of B cell depletion, but we didn't pursue that. At necropsy, the mice were indistinguishable by treatment type. So this table shows granuloma counts. There were no differences in the number or the size of the granulomas by the treatment group. I divided them into large, medium, and small and put them in the table here. And you can see that in both of the groups, there were mice that had different numbers of granulomas, and some of them were high, and many of them were low, and there wasn't any difference based on what treatment they got. Spleen granulomas, the same. We had a similar number of mice with and without splenic dissemination. So the impression from looking at these slides is that the disease is not quantifiably worse with the anti-CD20 treatment, but the granuloma histopathology is quite perturbed, which is what we're going to look at for the remainder of this. So these are PAS stained slides, and each of them, this is a 5x image that's been exported from a whole slide scan, and the value of this is I can show you pretty much a whole granuloma. The top is the anti-CD20 mice. The bottom are the isotype treated mice. The left-hand image is a mouse with a small number of spherules, and the right-hand image is a mouse with a large number of spherules. Generally speaking, the little red dots, and they're very hard to see at this low power, but I put them up here to show representatively that whether they've gotten the isotype or the anti-CD20, they could have a low number of spherules or a high number of spherules. So we actually counted these. I used QPath to help count the little red dots at obviously a much higher magnification than what you're seeing here. And there's no difference in the spherule count. You could see that the mean isn't particularly different between them. And if you look at the range, it's really hard to believe that they're different. I suppose if you ran a lot more mice, you might see a difference, but probably not. I think what's most dramatic about these slides, and this will be the focus of what we're going to mainly talk about, is that there's a profound loss of lymphoid aggregates in the anti-CD20 mice. You can see these are rather uniformly stained, kind of purplish red, whereas the isotype treated mouse have plenty of these dark purple, relatively round areas that consist almost very predominantly of lymphocytes. And we call these lymphoid aggregates. They may be called other things, but this is what we've been calling them since 2000. So this is dramatically different between these mice and the isotype mice. So the reduction and lack of lymphoid aggregates in the anti-CD20 mice is really the key histological feature between the two groups. These are higher powered images showing, again, there really is no lymphoid aggregate over here. And we have two of them in this particular image. The lymphoid aggregates are composed of T cells and B cells. And we did this work in like 2003 and showed by using an anti-CD3 stain and an anti-CD20 stain, that these are composed approximately half of T cells and B cells. And they actually sort of even split out a little bit in almost a half moon shape in the mice that we did originally. This has an anti-CD3 stain on it and is an even higher power to show you what we're talking about here. The T cells are not only just scattered here, you can see some of them are sort of trying to be together, but mostly where they stain, they're all over the place. And they're also greatly reduced, just observably reduced in number. Whereas down here in the isotype control mice, they're aggregated into their little spaces here, and the rest of these are going to be primarily B cells if you put an anti-CD20 stain on them. And then to fully demonstrate that the T cells are diminished in these mice, I counted them. We counted 520X fields per mouse and showed that there is, in fact, a statistically significant reduction in the T cells in the anti-CD20 treated mice. Finally, with the other half of the mice, we did the cytokine analysis, so there were 5 of each. And what we observed here out of the 13-plex is that the only thing that was different among these mice was the loss of the hallmark cytokines of coccidioidomycosis, Th1 and Th17 immune control, which drives the successful adaptive responses. And that is, we had almost no interferon gamma and almost no IL-17A found in these granuloma supernate, or these granuloma cultures, 24-hour cultures. And in the spleens, we actually didn't detect any cytokine differences. So I'm curious as to whether this is probably just a local effect in the lung granulomas, and it merits more study. So to summarize this, the loss of B cells at the time of infection distinctly perturbs primary granuloma formation. It changes the location of the T cells and reduces how many there are, and it greatly depresses the secretion of the controlling adaptive cytokines for cocci. However, the loss of the B cells did not affect 8-week survival, the lung granuloma numbers or their size, or the spheroidal counts on the slides. Now, this is only an 8-week study. It's a long model, and we do currently have a follow-up study running to assess 15-week survival. And on those mice, we will do organ fungal burdens to find out whether there is a difference in spheroidal burden in the lungs or whether there's greater dissemination to other organs across the body. Then it does look like there is a B cell role in lung granuloma formation, and we theorize that it's spatially sensitive or contact dependent based on the way these granulomas look. And the mechanisms may include MHC class II antigen presentation, which requires nearness, or T follicular helper cell maturation through PD-1, PD-L1, and we'd like to pursue some studies to evaluate these things in the primary granulomas. As usual, research is a team sport. Thanks to all our Valley Fever Center colleagues everywhere for helping. We have no conflict of interest. Thank you very much to the Cocci Study Group Program Committee for allowing me the opportunity to present our research. And a huge thanks to Amy Sue for scanning our slides for this study, because being able to quantitate whole slide scans is really a gift to being able to look deeper at some of these things in a quantitative manner. And also to Adam Buntsman for his analysis of the chronic granuloma RNA-seq data that set our feet on this path. Thank you.
Thank you. We have time for a couple of minutes, couple of questions.
Very nicely. So, well, as you know, so I think it was in the 90s, we published on human cocci nodules. Li-Jin Lin was the first author, and we noticed these. So we were mostly interested in cellular immunity, and that's what we were standing for. But we saw these lymphoid aggregates, and we thought, well, that's strange. And we looked back, you can see those in tuberculosis. They had never prior to that been described in coccidioidomycosis. And then again, we found that they were predominantly B cell, but also a mixture, just like in the mouse. And our theory was, well, maybe this is where antibody is made. And so this is seen also in human granulomata, but also I wonder if it would be reasonable to see if you could stain for antibody production. Maybe that's the site where antibodies occur in the granuloma. But certainly B cells do have a role in granuloma formation.
It could be. They're also, we didn't pursue it in this, but there are also a lot of plasma cells in a granuloma. And the worse the granuloma is doing, the more plasma cells there are, and they excrete tons of antibody. So, and they may be indicative of failing responses. That's what we think, that's what they look like in mice. Now, mice are not humans, but mice with failing responses have sheets and sheets of plasma cells, and many other things are reduced. However, what the B cells are doing very locally to modify the T cells, there are other things that can have MHC2 class presentation besides B cells that are in the granuloma and probably also doing this. But we were very surprised at the profound loss of interferon gamma and IL-17 in these. I was really surprised by it, frankly, because there are macrophages and all kinds of other things in there that may do presentation. So we've got a plan to try to look at the very local environment inside that granuloma. The other thing I forgot to say, it's a tertiary lymphoid organ. And that might have something to do with what things are doing inside of the granuloma that they're maybe not doing in the spleen, for instance. And we are really interested in this tertiary lymphoid organ and what's going on locally and why we can't kill the cocci in the granuloma and how it stays there, how the cocci stays there, why the granuloma stays there, and how we keep it under control at all. Thank you.
Do you want me to ask later? Are we doing it okay on time?
Well, could you talk later? Yeah,
you answered most of what I was going to ask, but I was also curious about the timing because the CD20 isn't going to deplete the plasma cells. So I'm curious if you do the infection first and vary the timing of the CD20.
CD20 doesn't deplete plasma cells, and we were not interested in plasma cells in this particular experiment. But when we do the chronic granuloma bulk RNA-seq, there's gobs and gobs of information that there are lots of plasma cells in it. We can talk about that later. Yeah. Thank you.
Thanks.
Okay. Thank you, Lisa. Okay, our second talk is Alison Harmon from Northern Arizona University. And her topic is identification of Coccidioides specific T cell clone in naturally exposed pigtails monkeys.